This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome, ocean explorers, to a journey beneath the waves. Today, we delve into the incredible world of chondrichthyes, the cartilaginous fish. From their ancient origins spanning hundreds of millions of years, to their unique anatomy, astonishing senses, and diverse life cycles, we'll uncover what makes sharks, rays, and chimeras true masters of the marine realm. Prepare to discover the secrets of these enduring and often misunderstood guardians of our oceans. Chapter 1. The Cartilaginous Enigma. Imagine a creature that has roamed the oceans for hundreds of millions of years, an ancient lineage of vertebrates that chose a path less traveled, an evolutionary divergence that continues to fascinate scientists today. We are talking about chondrichthyes, a class of animals whose very name, derived from the ancient Greek words chondros for cartilage and ichthys for fish, reveals their most defining characteristic, a skeleton primarily composed of cartilage, not bone. This fundamental difference sets them apart from the vast majority of fish species we know, the bony fish or osteichthyes, and underpins a myriad of unique adaptations that allow them to thrive in nearly every marine environment on Earth. For most of us, the idea of a skeleton immediately brings to mind hard, rigid bones. But chondrichthyes, a group encompassing the iconic sharks, the graceful rays and skates, and the mysterious chimeras or ghost sharks, challenge this norm. Their cartilaginous skeletons are far from simple or primitive. Rather, they represent a highly successful and evolved design. This skeletal choice offers a distinct set of advantages, primarily lightness and flexibility. A lighter body allows for greater buoyancy, reducing the energy expenditure needed to stay suspended in the water column. This inherent lightness also contributes to their remarkable agility, enabling swift movements and rapid changes in direction crucial for both predator and prey. The notochord, a flexible rod that provides skeletal support, is gradually replaced by a vertebral column during development in most chondrichthyes, but in holocephaly it remains intact throughout their lives, a subtle yet significant skeletal detail. Explain with examples, but do not use analogies that are not truthful. Consider the intricate structure of this seemingly simple material. Recent scientific explorations have delved into the nanoscale architecture of cartilaginous skeletons, particularly in species like the blacktip shark. What they've uncovered is far more complex than just soft tissue. This sharkitecture, as some researchers call it, reveals a sophisticated composite material. The mineralized cartilage in a shark's vertebrae, for instance, is not uniform. It comprises distinct regions, such as the corpus calcareum and the intermediale, both densely packed with collagen fibers and needle-like bioapatite crystals. These are the same mineral crystals found in human bones, but in sharks, they are intricately aligned with collagen strands in a highly ordered fashion. This precise arrangement creates porous structures reinforced by thick struts, allowing the skeleton to withstand mechanical strain from multiple directions. This resilience is vital for a creature constantly propelling itself through water, enduring repeated and multi-directional stress on its vertebral column. Perhaps most intriguing is the discovery of helical or spiral fiber arrangements woven within this mineral matrix. This layered spiral architecture acts as a formidable barrier, preventing cracks from propagating and escalating into catastrophic failures. Under mechanical stress, the collagen and mineral networks work synergistically, absorbing and dissipating force throughout the material. This natural design strategy, known as biomineralization, showcases how nature combines biological polymers with minerals to produce skeletal materials with outstanding mechanical properties. It's a blueprint for strength and flexibility that has persisted and been refined over hundreds of millions of years of evolution, offering compelling models for advanced material science and engineering today. Beyond the skeleton itself, the cartilaginous nature of these fish has profound implications for their internal systems. Unlike bony fish and other vertebrates, chondrichthyes lack bone marrow. This means they must produce red blood cells through alternative means, relying on specialized organs such as the spleen, the epigonal organ, a unique tissue surrounding the gonads, and, in some cases, Leydig's organ, which is found exclusively in cartilaginous fishes. However, highly specialized groups like the Chimeras or holocephali have lost both the Leydig's and epigonal organs, highlighting further evolutionary divergences within this class. Their hearts also feature a conus arteriosus, a muscular outflow tract that helps regulate blood flow, a characteristic anatomical detail. Furthermore, like all other jawed vertebrates, members of chondrichthyes possess an adaptive immune system, allowing them to mount specific responses to pathogens. Another critical consequence of their cartilaginous skeleton is the absence of a swim bladder, an air-filled organ that most bony fish use for buoyancy control. Without this internal flotation device, chondrichthyes employ other ingenious strategies to avoid sinking. Many species, particularly sharks, possess a remarkably large liver, sometimes making up to 20% of their body weight, which is rich in a low-density oil called squalane. This oil provides buoyancy, but it's often not enough on its own. Consequently, many sharks must keep swimming constantly to maintain their position in the water column, relying on the lift generated by their pectoral fins. The evolution of their appendages, particularly the pectoral and pelvic girdles, also showcases unique pathways. Originally, these girdles, which do not contain any dermal elements, did not connect. In later forms, each pair of fins became ventrally connected in the middle when scapulocoracoid and puboischiatic bars evolved, providing enhanced stability and maneuverability. The external covering of chondrichthyes also defies conventional fish scales. Instead, their tough skin is covered with dermal denticles, often referred to as placoid scales or skin teeth. These small tooth-like structures give their skin a sandpaper-like texture. In most species, these denticles are oriented in one direction, making the skin smooth when rubbed one way and rough the other. These dermal denticles provide not only protection, but also aid in streamlining, reducing drag as they move through water. This entire package of unique characteristics, from the mineralized cartilaginous skeleton and its implications for buoyancy and red blood cell production, to their specialized skin, illustrates how chondrichthyes have carved out an incredibly successful and enduring niche in marine ecosystems. Their ancient evolutionary journey, spanning hundreds of millions of years, has seen them persist through geologic upheavals. While their basic anatomy retains certain ancient elements, it also showcases a mosaic of highly evolved traits, demonstrating that the absence of bone is not a primitive lack, but a derived and highly adaptive evolutionary triumph. As we peel back the layers of what makes chondrichthyes so distinct, we begin to appreciate that their defiance of bony norms is not a limitation, but the very foundation of their extraordinary existence. But how did these ancient fish first emerge with such a unique blueprint, and what secrets does their deep past hold about the evolution of all jawed vertebrates? Chapter 2: Echoes from the Deep Past. Our journey into the world of chondrichthyes now takes us back through the vast corridors of time, millions upon millions of years, to uncover the deep evolutionary roots of these remarkable cartilaginous fishes. We must explore the echoes from their ancient past, a lineage that has navigated Earth's oceans for over 439 million years, persisting through cataclysmic changes and multiple mass extinctions. The story of chondrichthyes begins in the dim reaches of the Ordovician period, a time stretching back over 470 million years. While unequivocal fossils are scarce from this era, scientists have unearthed isolated scales made of dentine and bone that bear a striking resemblance to those of later chondrichthyans. These microscopic fragments, though their precise classification remains uncertain, offer tantalizing hints of stem chondrichthyans already present in these primordial seas, pushing their potential origins far deeper into geological history than once thought. However, the clearest and most unambiguous chapter of their tale opens in the early Silurian period, specifically around 439 million years ago, during the Aaronian stage. This is when the first undisputed fossils of Acanthiodean-grade cartilaginous fishes emerge from the ancient rocks of Guizhou, China. Discoveries like Chanodus and Fanjingshania represent not just early cartilaginous fish, but also the oldest known jawed vertebrates on Earth, and crucially, the oldest unambiguous gnathostome teeth. Imagine, these creatures were among the very first to possess a hinged jaw, a revolutionary adaptation that would transform the landscape of marine predation. Shortly after, around 436 million years ago, another early form, Shenacanthus vermiformis, sported thoracic armor plates, reminiscent of the armored plecoderms that would later dominate the seas. These Acanthiodeans, often referred to as spiny sharks, are now widely recognized not as a separate, distinct group, but as a paraphyletic assemblage, meaning they represent various branches leading directly to chondrichthyes. They are considered stem chondrichthyans, showcasing a mosaic of traits that bridge the gap between more primitive forms and the cartilaginous fish we know today. A pivotal example is the fossil Doliotis, which exhibits characteristics of both Acanthiodeans and chondrichthyans, offering a rare glimpse into this crucial evolutionary transition. This suggests that the first cartilaginous fishes evolved from ancestors much like Doliotis-like spiny sharks. By the dawn of the early Devonian, approximately 419 million years ago, the evolutionary experimentation with jaws had truly taken off. Jawed fishes had already diversified into three distinct groups: the armored placoderms, the burgeoning bony fishes, and the lineage encompassing spiny sharks and early cartilaginous fish. This period saw the appearance of Cladoceloche, the first abundant genus of shark, marking a significant milestone in their early diversification. Another key early unambiguous total group elasma branch, Phoebodus, emerged in the Middle Devonian, around 383 May, further illustrating the early radiation of true sharks. A fascinating aspect of this ancient lineage is that the cartilaginous skeleton, their defining feature, is not considered a primitive trait. Instead, research indicates it is a derived character, meaning chondrichthyes actually descended from bony ancestors. Over evolutionary time, their skeletons transitioned from bone to cartilage, a change that proved evolutionarily advantageous, contributing to their success in diverse ecological niches. The Paleozoic era, encompassing the Devonian, Carboniferous, and Permian periods, witnessed significant evolutionary radiations within the chondrichthyan lineage. Initially, diversity was relatively low in the Ordovician and Silurian. However, it saw a substantial increase in the early Devonian, primarily driven by the Acanthodons. As these stem chondrichthyans began to decline rapidly by the late Devonian, a second major radiation took hold in the earliest Carboniferous, led by the holocephalans, the group that includes modern chimeras. This period also saw the rise of diverse early sharks, including the C.T. enicanths, some of which grew to impressive sizes, comparable to modern great white sharks, around 7 meters, showcasing early large marine predators. This shift in dominance is thought to be linked to major environmental upheavals, such as the Hungenberg extinction event at the end of the Devonian. This event may have prompted holocephalans to expand their niches, dispersing into deeper water environments as other species faced displacement. Unraveling this ancient history is not without its challenges. The very nature of their cartilaginous skeletons, while advantageous for life in the water, presents a significant hurdle for paleontologists. Cartilage, being softer and more prone to decomposition than bone, rarely fossilizes in its entirety. This means that the fossil record of chondrichthyans is often fragmentary, composed predominantly of their durable teeth and dermal denticles, which are mineralized and much more resistant to decay. These small tooth-like scales provide invaluable clues about the morphology and evolution of extinct species. While entire articulated skeletons are rare, exceptional finds from localities like the late Jurassic Conservat Lagerstätten of southern Germany, such as the Solnhofen Archipelago and Nusplingen, have provided incredibly detailed insights into the cranial and postcranial morphology of ancient chondrichthyans. Modern techniques, such as computed tomography, CT scans, have further revolutionized the early chondrichthyan fossil record, allowing paleontologists to virtually reconstruct articulated remains and delicate brain cases, revealing hidden anatomical details previously inaccessible. Explain with examples, but do not use analogies that are not truthful. The evolution of key anatomical features also tells a story of deep time. For instance, the groundbreaking development of jaws, a characteristic shared by all chondrichthyes, is believed to have originated from the gill arches of their jawless ancestors. These supporting structures around the gills gradually transformed over millions of years into hinged jaws, dramatically expanding feeding capabilities and opening up new ecological roles. The debate continues as to whether their oral teeth evolved from dermal denticles migrating into the mouth, or if it was the other way around, or even if both had a common ancient origin. This is further complicated by the fact that early placoderms, other early jawed vertebrates, possessed sharp bony plates in their mouths rather than true teeth, highlighting the diverse evolutionary paths to oral processing. Regardless, the presence of these specialized structures has been a consistent feature throughout their history. Both major subclasses of chondrichthyes, the elasmobranchi, sharks, rays, and skates, and the holocephali, chimeras, have long and distinct evolutionary histories. Elasmobranchs are thought to have diverged from other jawed vertebrates around 420 million years ago in the Silurian. The fossil record of holocephali, although extensive, is also largely dominated by teeth, with their body forms often poorly understood for many extinct species. These early forms, such as the extinct Orodontiformes and Petalodontiformes, reveal a diverse array of ancient chimera relatives, some resembling skates or rays, others with beak-like jaws. It is important to note that the precise phylogeny of chondrichthyes, particularly the relationships between these groups and the exact definition of elasmobranchi, remains a subject of intense scientific debate, with morphological and molecular data sometimes presenting inconsistent pictures, reflecting the dynamic nature of paleontological and evolutionary research. As the Mesozoic era dawned, the evolutionary path of chondrichthyes continued its intricate course. While the very earliest members of the modern shark and ray group, known as Neosalaci, appeared in the Triassic, they truly began to diversify extensively from the early Jurassic onwards. This period saw the emergence of many of the modern orders of sharks and rays, coinciding with the decline of older groups like the hybodonts, which had been the dominant group of elasmobranchs during the Triassic and early Jurassic in marine environments, but persisted in freshwater ecosystems into the Cretaceous. Interestingly, another group of ancient sharks, the xenocanths, were abundant in both freshwater and marine environments for millions of years, highlighting the ecological versatility of early chondrichthyans.
This incredible journey through time, marked by the appearance of jaws, the evolution of a cartilaginous skeleton, and successive waves of diversification, underscores the enduring success of chondrichthyes. Their ancient lineage has proven remarkably resilient, adapting and thriving across vast geological timescales. But how did this deep evolutionary heritage shape the intricate sensory systems and anatomical marvels that allow them to navigate and dominate their aquatic worlds today? Chapter 3. Sensory Mastery and Anatomical Ingenuity. Having explored the fundamental nature of their cartilaginous skeletons and traced their ancient evolutionary journey, we now plunge deeper into the living world of chondrichthyes, uncovering the extraordinary sensory capabilities and ingenious anatomical designs that allow them to thrive in the vast, often dark, and complex aquatic realms. These are creatures that navigate their world by detecting faint electrical whispers, perceiving subtle pressure changes, and possessing bodies sculpted by millions of years of evolution, showcasing a biological mastery that continues to astound scientists. One of the most iconic and perhaps alien senses possessed by chondrichthyes is electroreception. Imagine navigating your world not just by sight, sound, or smell, but by detecting the faint electrical whispers of life around you. This is precisely what sharks, rays, and chimeras do, thanks to a remarkable network of jelly-filled pores known as the ampullae of Lorenzini. These specialized electroreceptors are scattered across their heads and snouts, acting like biological antennae. They can detect incredibly subtle electric fields generated by the muscle contractions of prey, even those buried beneath the sand or hidden in murky waters. This sixth sense is not merely for hunting. It also aids in navigation, allowing them to perceive the Earth's geomagnetic field like an internal compass, guiding their migrations across vast oceanic distances. Some research even suggests these ampullae can sense minute temperature gradients, providing another layer of environmental awareness. The evolutionary advantage of this sense is so profound that in certain species, like the hammerhead sharks, their distinct head shape is believed to have evolved to maximize the surface area for these electroreceptors, effectively widening their sensory net to locate prey with unparalleled precision. Complementing their electroreception is the lateral line system, a sensory network common to most fish. This system, composed of modified epithelial cells arranged along the sides of their bodies, detects motion, vibration, and pressure changes in the surrounding water. It provides an acute awareness of movements, whether it's the subtle displacement of water by a struggling fish or the approach of a larger predator, allowing chondrichthyans to perceive their aquatic environment in 3 dimensions, even in complete darkness. While electroreception and the lateral line offer unique insights into their surroundings, the more familiar senses are also highly refined. Chondrichthyans possess exceptionally powerful nostrils and olfactory organs, granting them an acute sense of smell. They can detect minute concentrations of chemicals in the water, such as blood from injured prey, over surprisingly long distances. This keen sense is a primary tool for locating food and often serves as the first alert to potential opportunities or threats. Their visual system, though sometimes overshadowed by their other extraordinary senses, is far from rudimentary. Most species have large, well-developed eyes, often equipped with a tapetum lucidum, a reflective layer behind the retina that enhances vision in low-light conditions, crucial for hunting in the dim depths or at night. While some species might rely less on sight in turbid environments, others, particularly active pelagic predators, employ their vision effectively. Even their hearing offers a distinct advantage. Their inner ears contain 3 large semicircular canals, essential for maintaining balance and orientation in the water column. While their sound detection apparatus has a limited range, it is particularly powerful at lower frequencies, allowing them to pick up the distant thrum of struggling fish or the low-frequency vibrations associated with large marine animals. The processing hub for all this sensory input is their nervous system. Chondrichthyans possess 8 to 10 pairs of cranial nerves, a fundamental component of their neural architecture. While their brains are often described as relatively simple, with the forebrain not greatly enlarged compared to some other vertebrates, they are surprisingly large in relation to their body size, sometimes approaching the relative weight of mammalian brains, and being about 10 times that of bony fishes. This doesn't necessarily mean higher intelligence in the human sense, but rather a capacity for complex integration of their specialized sensory data, achieved with a lower density of nerve cells, making it less energy demanding. However, diversity exists even here. The primitive megamouth shark, for example, has a brain of only 0.002% of its body weight. Furthermore, the structure and formation of myelin in their nervous systems are almost identical to that of tetrapods, suggesting that chondrichthyans were a pivotal group in the evolutionary timeline of myelin development.
Beyond their sensory prowess, the anatomical ingenuity of chondrichthyes is equally remarkable, particularly in how they manage essential life functions like respiration and digestion without the structures found in bony fish. Their respiratory system, for instance, is characterized by 5 to 7 pairs of gill slits that open directly to the exterior, lacking the bony operculum that covers the gills of osteichthyes. The method of oxygen uptake varies significantly with their lifestyle. Pelagic species, those that cruise the open ocean, often rely on ram ventilation. They must keep swimming forward to force oxygenated water over their gills. In contrast, many demersal species, those that dwell on or near the seafloor, have developed spiracles, small openings located behind each eye. These spiracles, which can range from tiny and circular, such as found on the nurse shark Ginglimostoma serratum, to extended and slit-like, as seen on the wobbegongs Orectolobidae, allow them to actively pump water in and over their gills while stationary, preventing their gills from becoming clogged with sand or debris when resting on the bottom. However, many larger, fast-swimming pelagic species, such as mackerel sharks and thresher sharks, have lost their spiracles, relying solely on their constant movement.
Their digestive system, too, exhibits unique adaptations for efficiency. Lacking the long, coiled intestines typical of many vertebrates, chondrichthyes possess a relatively short intestine that contains a distinctive internal structure called a spiral valve. This corkscrew-like fold dramatically increases the internal surface area for nutrient absorption, compensating for the shorter length and ensuring efficient digestion of their often protein-rich diets. With the exception of chimeras, most chondrichthyes also possess a cloaca, a single posterior opening that serves as the exit point for digestive, urinary, and reproductive systems.
The design of their appendages further highlights their specialized adaptations for aquatic life. The heterocircle tail, a primary characteristic of most sharks, features an elongated upper lobe that provides powerful thrust and propulsion through the water. In rays, the pectoral fins have undergone a radical transformation, expanding laterally and fusing with the head and trunk to form their characteristic flattened disc-like bodies. This allows them to fly through the water with graceful wave-like motions or to lie camouflaged on the seabed. The skeletons of rays are notably more complex than those of sharks, with attachments of the pectoral girdle to the vertebral column and the anterior basals of the pectoral fins to the skull or neurocranium. Furthermore, the vertebral column between the cranium and pectoral girdle is often fused into a tube. This intricate arrangement serves to support the broadened pectoral fins and to resist their propulsive thrust. Many sharks and all chimeras also possess 2 dorsal fins, sometimes armed with stiff spines. In some chimeras, these dorsal fin spines can even be venomous, offering a potent defense mechanism. The chimeras present a fascinating case of further anatomical divergence within the chondrichthyes. Unlike their elasmobranch relatives, chimeras possess a single gill opening on each side, covered by a soft operculum, a feature more reminiscent of bony fish. Their upper jaws are completely fused to their cranium, a condition known as autostylic suspension, which differs significantly from the more flexible jaw articulations seen in sharks and rays. Adult chimeras also lose most of their dermal denticles, retaining them primarily on specialized clasping organs found on the head and pelvic fins of males, which are uniquely adapted for reproduction. Their mouths are small apertures surrounded by lips, giving them a distinctive parrot-like appearance. And their teeth are fused into continuously growing tooth plates, perfectly adapted for crushing the mollusks and other invertebrates that make up their diet. They lack a true stomach, with their gut simplified, and the stomach merged with the intestine. Their long, thin tails, which often resemble a rat's tail, contribute to their unique deep-sea profiles.
The anatomical and sensory marvels of chondrichthyes are not isolated features. They work in concert, forming a highly integrated system that enables their survival and success. From the subtle electrical detection of prey to the efficient hydrodynamics of their bodies and the specialized adaptations of their internal organs, every aspect of their biology is finely tuned to their aquatic existence. This intricate interplay of senses and form allows them to navigate, hunt, and reproduce in a world that remains largely hidden to human eyes. Indeed, it is their ability to perceive and interact with their environment in such diverse and sophisticated ways that has ensured their enduring presence in marine ecosystems for hundreds of millions of years. This mastery of their environment extends profoundly to their life cycles, particularly their diverse and often elaborate reproductive strategies, which we will explore next.
Chapter 4. Reproductive Wonders. Diverse beginnings from the ancient depths of their evolutionary past to the intricate workings of their senses, chondrichthyes have proven themselves masters of adaptation. Yet, perhaps nowhere is their unique biology more strikingly displayed than in their diverse and often elaborate reproductive strategies. Unlike the vast majority of fish that release countless eggs and sperm into the water column, hoping for chance encounters, cartilaginous fish employ a suite of sophisticated methods, all rooted in a fundamental biological decision: internal fertilization. This critical step, common to all living chondrichthyes, sets the stage for a remarkable spectrum of life's beginnings, from protective egg cases to live births that rival those of amniotes.
The journey begins with the male, equipped with specialized copulatory organs known as claspers. These are modified portions of the pelvic fins, often cigar-shaped, hemipenis, and are grooved structures designed for the precise transmission of sperm into the female's cloaca, or, in the case of chimeras, their separate urogenital opening. This internal union ensures a higher probability of successful fertilization compared to external spawning, a strategy that has undoubtedly contributed to the long-term success of these animals. But the ingenuity doesn't stop there. In some groups, particularly the enigmatic chimeras or ghost sharks, the males possess even more elaborate structures to ensure mating success. Beyond the standard pelvic claspers, a male chimera may feature an unpaired frontal tentaculum on its forehead and paired pre-pelvic claspers, all adorned with hook-like dermal denticles. While primarily used for firmly grasping the female during copulation, in some species, like the elephant fish, Calorhynchus mealy, the frontal tentaculum also aids in finding food, showcasing a remarkable dual purpose. These additional clasping organs are a testament to the evolutionary pressures driving reproductive security in the deep, dark ocean environments many chimeras inhabit. Once fertilization is internal, the path to development diverges into three primary modes: ovoparity, where eggs are laid externally; ovoviviparity, where eggs hatch inside the mother and live young are born; and viviparity, a rarer form involving direct maternal nourishment and live birth. Each strategy comes with its own set of advantages and challenges, reflecting millions of years of evolutionary fine-tuning.
Ovipary, or egg-laying, is a fascinating display of biological engineering. The eggs of oviparous chondrichthyans are encased in a protective structure often referred to as a mermaid's purse. These cases are typically made of collagen protein strands, giving them a rough, leathery texture. They are far from simple shells. Their designs are remarkably varied and often species-specific, showcasing adaptations to protect the developing embryo from predators and environmental hazards. Most egg cases are roughly rectangular, with distinctive projections known as horns at each corner. Some species further enhance this protection with a fibrous material covering the outside, aiding in attachment to the substrate, while others are smooth or bumpy. It is worth noting that ovipary is completely absent in the superorder Squalomorphae. The formation of these cases is a marvel in itself. Shortly after internal fertilization, the fertilized ovum enters a partially formed egg case within the female's oviduct. The rest of the case then forms around the ovum, and once complete, it is deposited outside the body. Typically, egg cases are produced in pairs, each containing a single fertilized embryo. However, exceptions exist, such as the big skate, whose large egg cases can house up to 7 embryos, though usually 3 or 4 are found. The deposition sites are carefully chosen, often in sheltered environments like kelp forests or rocky seafloors, where the cases can be anchored or hidden. Gestation periods within these cases can be remarkably long, ranging from a few months to over a year, depending on the species. After a week or two post-deposition, small slits often open on the sides of the case, allowing water to flow through and the embryo to fan its tail, promoting essential gas exchange with the surrounding seawater. The diversity of these egg cases is truly astonishing. Consider the bullhead sharks, members of the order Heterodontiformes. Their egg cases are distinctively auger-shaped, featuring prominent spiral flanges. This unique design allows them to be wedged securely into the crevices of rocky seafloors, providing protection from predators. Some bullhead sharks, however, have been observed depositing their eggs on sponges or seaweed. The hatchlings emerging from these cases are unusually large for sharks, sometimes exceeding 14 centimeters in length, a significant head start in a dangerous world. Female Japanese bullhead sharks even exhibit a degree of social behavior, depositing their eggs in communal nests. In contrast, the egg case of the Mexican horn shark displays tendrils and more rigid flanges, suggesting an evolutionary shift towards anchoring rather than wedging. Other oviparous sharks also showcase specialized designs. Bamboo sharks and zebra sharks, part of the carpet shark order, lay oval egg cases covered with adhesive fibers, which serve to securely fasten them to the seafloor. Some ground sharks, notably certain cat sharks, produce purse-shaped egg cases equipped with long tendrils at their corners. These tendrils act as natural anchors, snagging onto structures on the seabed to prevent the cases from being swept away. The size of these cases can vary dramatically, from the small spotted cat sharks' cases, around 5 centimeters long, to the greater spotted dogfishes, which can reach 10 centimeters, excluding their lengthy tendrils. Skates are the only group of rays that are oviparous, depositing their egg cases onto the seafloor after internal fertilization. These cases possess distinguishable characteristic traits unique to each species, making them valuable tools for identification. Features like the presence or absence of a fibrous covering and the keel, a flexible structure running laterally along the outer edge, are key. Some species have broad keels, others narrow, and some keels even extend the length of the horns. The big skate, as mentioned, produces particularly large, smooth, and convex egg cases, with broad keels, often containing multiple embryos, a rarity among cartilaginous fish. The long-nosed skate, while also producing large cases, typically encloses a single embryo within its brown, externally fibrous shell. Even chimeras, the deep-sea ghost sharks, are oviparous, producing egg cases that are spindle or bottle-shaped, complete with lateral fins. These are laid on the seafloor, and their bulbous centers, flanked by flattened collagen tissue that forms a tail-like anterior end, contribute to their unique appearance. Sharp projections on both ends of the case further aid in securing them between rocks and deterring potential predators.
Evidence from the fossil record also hints at the ancient origins of these elaborate egg-laying strategies, with extinct chondrichthyans like hybodonts and xenocanths producing egg cases such as Paleoxyrus and Phaeolia, some of which bore spiral collarettes and were found in freshwater environments, a stark contrast to many modern species. Despite their robust design, these egg cases are not invulnerable. Predation by gastropods, which bore into the exterior, and even other sharks remains a significant source of mortality for the developing embryos.
Moving beyond external egg-laying, the most common reproductive strategy among sharks is ovoviviparity, sometimes referred to as aplacental viviparity. In this mode, the fertilized eggs are retained within the mother's oviduct, where they hatch internally. The developing embryos are nourished primarily by a yolk sac, much like those in externally laid eggs, but they benefit from the enhanced protection of developing within the maternal body. While there is no direct placental connection for nutrient transfer, the mother provides a stable, protected environment. This strategy results in live birth, with the young emerging fully formed and ready to face the world. A dramatic and rare variation of ovoviviparity, known as intrauterine cannibalism, occurs in certain species such as the sand tiger shark. In this gruesome but effective strategy, the first developed embryo in each of the mother's 2 uteri consumes its less developed siblings, as well as any unfertilized eggs, ensuring that only a few exceptionally robust and well-fed pups are born. This extreme form of sibling rivalry guarantees that the surviving offspring are highly competitive from the moment of birth. True viviparity, or placental viviparity, where the embryos receive direct nourishment from the mother through a placenta-like connection, is a rarer, more advanced form of reproduction among chondrichthyes, akin to that seen in many amniotes. In this scenario, the mother actively transfers nutrients to her developing young, allowing them to grow larger and more robust before birth. Species like the great hammerhead shark exhibit this form of reproduction, delivering live, well-developed young that have received sustained maternal support throughout their gestation. For instance, the daggernose shark, Isogomphodon oxyrhynchus, exemplifies this strategy. Females live to approximately 20 years, while males live to about 12. Females are thought to give birth on alternate years to litters of 1 to 8 pups, typically during the rainy season in Brazil, January to June, after a gestation period of 12 months. This strategy further enhances the survival rate of individual offspring, compensating for the typically small litter sizes. Regardless of the specific mode, chondrichthyes generally exhibit long gestation periods. Some species have pregnancies lasting up to 2 years, a duration longer than any other vertebrate. This extended developmental time, coupled with the internal protection afforded by ovoviviparity and viviparity, means that the young are born or hatch at a relatively large size and a more advanced stage of development. This life history strategy, characterized by producing fewer, larger, and more capable offspring, stands in stark contrast to the vast numbers of small, vulnerable young produced by most bony fish. However, a crucial trade-off of this strategy is a slower reproductive rate and a longer time to reach sexual maturity. The Greenland shark, for instance, a colossal inhabitant of Arctic waters, does not reach sexual maturity until it is an astonishing 156 years old, highlighting the extreme end of this slow-paced reproductive cycle. A concerning phenomenon linked to these reproductive patterns is capture-induced parturition, a term encompassing both premature birth and abortion. This occurs frequently in sharks and rays when they are caught during fishing activities. The stress of capture can trigger the premature expulsion of developing young or egg cases. This event is often mistaken for natural birth by recreational fishers and is rarely accounted for in commercial fisheries management, despite its documented occurrence in a significant number of live-bearing species. This oversight can lead to underestimation of fishing impacts on populations already vulnerable due to their inherently slow reproductive rates and long maturation times.
Explain with examples the diverse reproductive strategies of chondrichthyes, from the intricately designed mermaid's purses to the varied forms of live birth, are a cornerstone of their evolutionary success. They represent a significant investment in individual offspring, prioritizing survival and developmental advantage over sheer numbers. This careful approach to life's intricate beginnings has allowed them to endure for hundreds of millions of years, navigating the complexities of marine ecosystems. But these very adaptations, which have ensured their longevity, also render them uniquely susceptible to external pressures, particularly those imposed by human activities. What then is the critical ecological role these ancient mariners play? And what challenges do they face in an increasingly human-dominated ocean?
Chapter 5, Guardians Under Threat. Welcome back, ocean explorers. We've journeyed through the evolutionary marvels and intricate biology of chondrichthyes, uncovering the secrets of their cartilaginous skeletons, their extraordinary senses, and their diverse reproductive strategies. Now, as we reach the final chapter of our exploration, it's time to confront a pressing reality: the vital, often unseen role these animals play in maintaining the health of our oceans and the profound crisis they currently face. These ancient mariners are far more than just fascinating creatures. They are, in many respects, the guardians of the ocean, and their disappearance would trigger catastrophic changes across marine ecosystems. They are often considered keystone species within their habitats. Their presence, often as apex predators, is crucial for maintaining the delicate balance of an entire ecosystem. Imagine a complex web of life in the ocean. These animals sit at the top, influencing the populations of species below them in the food chain. For instance, the great white shark, a formidable predator, helps to keep populations of seals and other large marine mammals in check. This control prevents overgrazing of certain prey species, which could otherwise deplete resources for others. Without these top predators, a phenomenon known as a trophic cascade can occur, where the removal of one species triggers a domino effect of ecological imbalances throughout the entire food web. Prey species can proliferate unchecked, leading to the collapse of lower trophic levels, ultimately reducing the overall biodiversity and resilience of the ecosystem.
Rays, too, play a critical, albeit different, ecological role. Many species with their flattened bodies are adapted for a demersal lifestyle, feeding on benthic organisms such as small fish, crustaceans, and mollusks buried in the seafloor. By foraging in these habitats, they contribute significantly to nutrient cycling. Their movements stir up sediments, releasing trapped nutrients that then become available for other marine life, including plants and smaller invertebrates. Stingrays, for example, aerate the seafloor as they hunt, benefiting plant growth. This constant interaction ensures a dynamic and healthy sediment environment. Skates and rays generally support biodiversity within their habitats, serving as a food source for larger predators and promoting a stable ecosystem capable of withstanding environmental changes. The diverse feeding habits, from the filter-feeding whale shark to the crushing jaws of many rays, illustrate their varied contributions to oceanic health. However, despite their immense ecological importance, chondrichthyes are currently facing an unprecedented crisis, primarily driven by human activities. The statistics are stark. In just 7 years, the fraction of chondrichthyine species threatened with extinction has jumped from a quarter to a third. A recent reassessment indicates that 391 out of 1,199 species, or 32.6%, are now classified as threatened according to international conservation criteria. This estimate is considered conservative as it classifies all species for which there is insufficient data as non-threatened, a presumption that is unlikely to be accurate. If these data-deficient species were as threatened as those we have assessed, the overall percentage could rise to as high as 37.5%. The IUCN Red List Index, RLI, a tool used to track extinction risk, reveals a grim trend. The extinction risk for chondrichthyans has worsened by 19% in the past 50 years. This makes them the most threatened group of vertebrates after amphibians, a truly alarming statistic. The dominant threat impacting all 391 threatened species is overfishing. For decades, these animals have been caught, both intentionally and unintentionally, at unsustainable rates. Globally, countries like Indonesia, Spain, and India lead in shark fishing, followed by Mexico and the U.S., highlighting the widespread nature of this pressure. Moreover, harmful fishing subsidies in some countries exacerbate the risks to these populations. The global catch of sharks and rays, for instance, doubled from 750,000 metric tons to 1.5 million metric tons between 1950 and 2000, with fishing efforts tripling. This relentless pressure has had devastating consequences, halving their populations since 1970. More than 100 shark species, for example, saw their populations decline by over 80% between 1975 and 2009 alone. One of the most widely recognized and often condemned practices is shark finning, driven by the demand for shark fin soup in some cultures. While the demand for shark fin soup has seen some decline since the early 2000s, this has not necessarily reduced mortality rates. International agreements have helped regulate the fin trade, but often the animals are now retained and new uses for their parts are found. For instance, the trade in shark meat doubled in value from $157 million in the early 2000s to $283 million in 2016, now valued at 1.7 times that of the fin trade. Deep-sea sharks are also targeted for their liver oil, which has commercial applications. Species like the school shark, gulper shark, and basking shark, all of which are assessed as vulnerable due to overfishing, have been particularly impacted by this demand. Beyond targeted fishing, bycatch remains a significant problem. Chondrichthyans are frequently caught unintentionally in fisheries targeting other species through methods like trawling, gill netting, and longlining. Even if they are not the primary target, the stress of capture can lead to injury or death. Their reproductive strategies make them particularly vulnerable to such pressures. For instance, the giant manta ray does not begin reproducing until it is around 8 to 10 years old, and then gives birth to a single pup only once every 3 to 4 years. This slow reproductive rate means they have very little capacity to recover from sustained fishing pressure, making their populations highly susceptible to decline. A concerning trend observed in some populations, such as the small spotted cat shark in the Mediterranean, is a decreasing trend in maturity, suggesting a potential evolutionary response to overfishing, where individuals are reproducing earlier but possibly with fewer offspring, further impacting population viability. Habitat loss and degradation represent another severe threat. Coastal development, pollution, and destructive fishing practices like bottom trawling directly impact the critical habitats that chondrichthyes rely on for feeding, breeding, and nursery grounds. These activities can destroy delicate ecosystems such as mangrove forests and coral reefs, which are vital for the survival of many juvenile sharks and rays. The Mediterranean Sea, a semi-enclosed basin with intensive fisheries and high human population density, has become a key hotspot of extinction risk for chondrichthyans. Of its 89 species, 49 sharks, 38 batoids, 2 chimeras, over half, 39 out of 73 assessed species, are considered threatened, including 20 critically endangered, 11 endangered, and 8 vulnerable. Local extinctions of species like the flapper skate and certain sawfish and angel shark species have already been observed in this region. The dagger-nosed shark, a species with one of the smallest ranges of any elasma branch, is now listed as endangered under the Endangered Species Act and CITES Appendix 2, primarily due to bycatch in unregulated artisanal gillnet fisheries. Climate change and ocean acidification also contribute to the crisis, albeit sometimes as less dominant threats compared to overfishing. Rising ocean temperatures can disrupt migration patterns and breeding cycles, while ocean acidification can impact the calcification processes of marine organisms lower in the food chain, ultimately affecting the food sources available to chondrichthyans. Public perception, often shaped by sensationalized media portrayals, has historically hindered conservation efforts. Depictions of sharks as dangerous, mindless killers, exemplified by movies like Jaws, have fostered fear and led to culling practices that further decimate populations. This negative narrative often overshadows their critical ecological contributions. The consequences of this crisis are far-reaching. The decline in chondrichthyan populations is not merely about the loss of individual species. It signifies a significant drop in the functional diversity of marine ecosystems. A 22% drop in functional diversity has been observed, reflecting the loss of the unique ecological roles these animals perform. This loss compromises the resilience and stability of ocean environments, making them more susceptible to other disturbances. The Java stingray, for instance, became the first marine fish confirmed to have gone extinct directly due to human activity, a grim testament to the current trajectory. However, there is hope, and significant conservation efforts are underway globally. Research and monitoring are crucial to filling the vast data gaps that still exist, particularly for those data-deficient species whose true conservation status remains unknown. Scientists are working to understand population dynamics, reproductive cycles, and habitat use to inform more effective management strategies. Implementing sustainable fishing practices, reducing bycatch through improved gear and techniques, and establishing and enforcing marine protected areas are all vital steps. Habitat restoration efforts, such as replanting mangroves and coral, aim to rebuild critical nursery and feeding grounds. Public awareness and education campaigns are working to shift the narrative, highlighting the ecological importance of these animals and promoting their protection. International agreements and conventions, such as CITES, aim to regulate the trade of threatened species, while organizations like the International Union for Conservation of Nature, IUCN, continue to assess and classify species, providing the scientific basis for conservation action. Beyond their ecological value, chondrichthyes also hold economic significance. While fisheries have historically exploited them, increasingly ecotourism, particularly shark diving, demonstrates their economic value alive rather than dead. Furthermore, byproducts from these animals, such as collagen and glucosamine from cartilage used in health supplements, cosmetics, and wound healing, or chondroitin sulfate for treating joint conditions, and vitamins A and D from liver oil, popular as nutritional supplements, have various industrial and medicinal applications. Shark skin is also used in specialized leather products. The challenge lies in ensuring that any utilization of these resources is sustainable and does not further imperil already vulnerable populations. The story of chondrichthyes is one of ancient resilience meeting modern challenges. These creatures, whose evolutionary journey spans epochs, are now at a critical juncture. Their survival depends on a global shift in human behavior, a recognition of their indispensable role as guardians of the ocean, and a concerted effort to implement adaptive management and conservation strategies. Can we, as a global society, learn to coexist with these magnificent animals, ensuring their continued presence in the intricate tapestry of marine life for generations to come? Or will their ancient lineage finally succumb to the pressures of the Anthropocene? From their ancient cartilaginous origins to their sophisticated senses and diverse reproductive strategies, chondrichthyes represent an extraordinary evolutionary success story. These sharks, rays, and chimeras are not just fascinating. They are keystone species, vital for maintaining ocean health. Yet, these ancient mariners face an unprecedented crisis, primarily from human activities like overfishing, pushing over a third of species towards extinction. Their slow reproductive rates make them uniquely vulnerable. The fate of these ocean guardians now rests in our hands. Can we secure their future and, in doing so, safeguard the health of our planet's oceans? Thank you for listening.